Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will talk about catalase test. Catalase is an enzyme, and catalase test detect whether a bacteria carries catalase enzyme or not. So, catalase test detects catalase enzyme. And most commonly, catalase test is used to differentiate between staphylococci, which are catalase positive, from streptococci and enterococci, both are catalase negative. Most commonly, this test is used to differentiate between staphylococci from streptococci and enterococci. Both of these are catalase negative, whereas staphylococci are catalase positive. That means they carry catalase enzyme with them, whereas streptococci and enterococci do not carry catalase enzyme with them. Just remember, staphylococci, streptococci, enterococci, three of them are gram-positive cocci. To differentiate between them, catalase test is used. But the question is why this enzyme is so important in the identification of staphylococci and streptococci and how it plays its role. Or what is the principle of this test? So, facultative anaerobes and aerobic bacteria during aerobic respiration, at the end of aerobic respiration, electron is produced. This electron is accepted by oxygen. So oxygen is acting as electron acceptor over here. Oxygen molecule will accept this electron and resulting in the formation of hydrogen peroxide and water, H2O. I'm just repeating it. Aerobic bacteria and certain facultative anaerobic bacteria, at the end of aerobic respiration, electron is produced which is accepted by oxygen and resulting in the formation of hydrogen peroxide and water, right? This hydrogen peroxide is very lethal for bacterial cellular structure and enzymes. So this hydrogen peroxide is converted into non-harmful products. Hydrogen peroxide is converted into water and oxygen with the help of catalase enzyme. So catalase enzyme is protecting staphylococci from the dangerous effect of hydrogen peroxide. As I have already mentioned, this catalase test is used to differentiate between staphylococci from streptococci, but there are other bacteria which are also catalase positive. First of all, staphylococci are catalase positive. Second, E. coli. Third, Pseudomonas. And Listeria, Nocardia. Also, Aspergillus, which is a fungus, is also catalase positive. Talking about how to perform this test and what is the procedure of this test, first we'll discuss what are the material and apparatus required. In this test, we need 3% hydrogen peroxide. This hydrogen peroxide is light sensitive, so it is stored in brown bottles. Other than 3% hydrogen oxide, we need slides, test tubes, and a wooden stick. Now, talking about the procedure of catalase test, there it can be performed by two ways. First, it can be performed by slide method, and catalase test can also be performed by test tube method. In the slide method, we need a clean glass slide, right? On a clean glass slide, we'll put a drop of water in the center, right? Then from the agar plate, we will pick a bacterial colony, and with the help of a wooden stick, right? We're not using a wire loop over here because uh, iron can result in the oxidation redox reaction and is giving a false positive result. So we will use a wooden stick in this method. We'll pick a colony of bacteria, we'll mix this with the water. Then in the third step, we will add few drops of hydrogen peroxide. When we'll add the drops of hydrogen peroxide, on the slide we can clearly see bubble formation, because this hydrogen peroxide which we have added, bacteria use this catalase enzyme to break it down and resulting in the formation of oxygen, which can be seen in the form of bubbles. So in slide method, we took a clean glass slide, put a drop of water, mix bacteria, mix bacterial colony, and then add few drop of hydrogen peroxide. Bubble formation will tell us that the test is positive. If there is no bubble formation within 5 to 10 seconds, it means the test is negative. And this bubble formation is due to the production of oxygen. The second method is test tube method. In this method, we will take a test tube, we will add few drops of hydrogen peroxide in the test tube. Then we will pick a bacterial colony from the agar with the help of a wooden stick, right? Then, then we will throw this stick into the test tube, and then we will cover the test tube with the test tube cap. We will see that there will be a bubble formation, vigorous bubble formation. A large amount of bubbles will be formed. That will tell us the result is positive. So in test tube method, take a test tube, add few drops of hydrogen peroxide, pick a colony of bacteria, throw the wooden stick into test tube and cover the test tube. with cap, and bubble formation will tell us that the result is positive and the test is positive. If we briefly describe catalase test detects catalase enzyme. This test is most commonly used in lab to differentiate between staphylococci, which are catalase positive, from streptococci and enterococci, which are catalase negative. Catalase enzyme can convert hydrogen peroxide into water and oxygen. This oxygen is seen in the form of bubble formation. When we perform this test, this can be done by two ways: slide method and test tube method. In the slide method, we'll take a clean glass slide, put a drop of water, then we'll mix the bacterial colony with the water, then add a few drops of hydrogen peroxide. Bubble formation will clearly be visible if the test is positive within 5 to 10 seconds. But this slide method is a biosafety concern, because due to this bubble formation and aerosol formation, these bacteria can be inhaled by the person who is performing this test. So it is ideally best to perform test tube method. In this method, we'll take a clean glass slide, we'll add few drop of hydrogen peroxide, then with the help of a wooden stick, we will pick a bacterial colony, we will mix with the, we'll just throw the stick into the test tube and cover the mouth of the test tube. Within 5 to 10 seconds, bubble formation will be seen due to oxygen production. That indicates the test is positive. That's it from this video. If you have any question regarding this topic, please ask in the comment section. For more microbiology content, please like, share and subscribe to my channel. Thank you.